Okay, let's dive right into this BioinfoBytes explainer. Today, we are taking a high-speed cinematic journey from the microscopic to the macroscopic. Our goal? To understand one of the absolute most powerful tools in modern genomics, the DNA microarray. We're going to visualize this data in continuous motion, so lock in, because it moves fast.
Right off the bat, I want you to imagine shrinking an entire massive genomics laboratory down to a single 1 square centimeter chip. It sounds like science fiction, right? But this tiny piece of glass or silicon actually allows us to simultaneously measure the expression of 50,000 genes in a single pass. So we're not just taking a step forward in biology here, it is a massive fundamental leap in parallel data processing.
Section 1. Chip design and probes. Let's look at the hardware.
To comprehend how we extract that much data, we really need to rapidly zoom in on the physical hardware of the chip itself. Using advanced Affymetrix multi-probe photolithography, millions of short, covalently attached oligonucleotide sequences are etched directly onto a glass wafer. These oligos are usually between 25 to 70 nucleotides long, and they act as dedicated complementary probes. And the wild part is, every single microscopic spot on this grid corresponds to exactly one target gene.
So you might be wondering, how does this ultra-dense grid actually work? Well, it entirely relies on one beautiful fundamental biological principle: nucleic acid hybridization. Because DNA strands are naturally complementary, our unknown target samples will naturally seek out and physically lock onto their exact chemical match on the board. They bind tightly by forming hydrogen bonds. If there's a match, they stick. If not, they just wash right off.
Now, what's really interesting here is the relentless, rapid-fire procedural pipeline. First up, we isolate the messenger RNA from our biological sample. But because RNA is fragile, we convert it into complementary DNA, or cDNA, and tag it with fluorescent dyes. We then apply this glowing mixture directly onto the microarray plate for hybridization. Let it bind, wash off the excess, and hit it with a rapid laser scanner for detection.
Section 2. The two-color hybridization cascade. This brings us to a brilliantly color-coded battle taking place at the microscopic level. In a two-color array, we label our treatment sample with an electric green dye called C3. Then, we label our healthy control with a coral red dye called Cy5. As they compete and bind to the chip, the resulting colors tell us everything we need to know. An electric green spot? That means the gene is upregulated in the treatment. Coral red means it's upregulated in the control. An amber or yellow spot, that's a tie, equal gene expression in both. And a completely dark spot means that specific gene is turned off in both samples. Section 3. Digital translation, laser scanning, and data analysis.
With the hybridization battle complete, we zoom out slightly. A precision laser sweeps across the slide, exciting those molecular dyes. A high-resolution camera then instantly captures this glowing grid, calculating the raw luminescence intensity for every single dot on the chip. What you're left with is the raw, visual fingerprint of a living organism's active genome.
But we definitely can't stop at just raw intensity images. Complex mathematical models step in to log transform and normalize the data. Think of it like tuning a radio to the right frequency to filter out all that systemic background noise. From there, algorithms generate massive clustering heat maps and volcano plots, revealing the hidden, dynamic relationships between thousands of genes all at once.
Section 4. Real-world medical applications.
Finally, zooming all the way out to the human level, why does reading this massive grid actually matter for us? Because this massive parallel processing is literally revolutionizing modern healthcare. We use these arrays to instantly classify complex cancers by detecting mutations in tumor suppressor genes like P53 and BRCA1. We use them for personalized drug response profiling and pharmacogenetic screening, like analyzing the CEPA450 gene to see exactly how a patient will respond to a specific pharmaceutical drug. And we also use them for rapid SNP genotyping arrays. It takes the guesswork entirely out of medicine.
We have evolved from studying a single gene over months of painstaking lab work to capturing an organism's entire transcriptomic universe in one continuous, glowing sweep. It's an absolutely massive leap forward, and it leaves us with one provocative question to end on. 50,000 genes, one chip. What life-saving pattern will you decode next?